All right, welcome back to another video for Module 5. This is the second to the last video where we are going to be talking about cellular respiration. So we've talked a lot about energy, what energy is used for, metabolism, all that good stuff. Now we're going to finally talk about how ATP is made. So how we take the molecules from our food, so those organic molecules like glucose, and our cells convert that molecule into ATP, which will then be used to fuel a lot of our metabolic processes. And so cellular respiration actually has several different steps for that process. And we're going to talk about each of the steps independently, and then we'll summarize them all at the end of the video. And so the first process, the first step to cellular respiration is glycolysis. Glycolysis is where our cells take a glucose molecule, and a glucose molecule is the sugar in which heterotrophs get our energy, right? So we consume other organic molecules to get these glucose molecules, and then the glucose molecule is converted into ATP through the process of cellular respiration. And glycolysis is that first step in breaking down that glucose molecule for energy used in metabolism. Glycolysis actually takes place in the cytoplasm of prokaryotic cells and eukaryotic cells. Okay. After glycolysis happens, the products then travel to the mitochondria, where the rest of the cellular respiration process occurs. So here we have our basic image of glycolysis and the different steps. And what we're going to do is we're going to kind of walk through this a little bit. So glycolysis in the cytoplasm begins with a glucose molecule. Remember, glucose is 6 carbons, forms a ring structure, and the process of glycolysis actually uses 4 ATP molecules. So 2 ATP molecules are used to split glucose into glyceraldehyde 3-phosphate, which is shown here. So 2 3-carbon molecules, and then an NAD plus molecule takes the electron or the proton off of these molecules to make NADH. Now, NADH is going to be very important because they are the molecules that's going to drive the process of the last part of the cellular respiration cycle. So that oxidative phosphorylation step, which is also known as the electron transport chain. And so we've used 2 ATP to break down glucose into 2 3-carbon molecules. Then those molecules make NADH, and we use 2 ATPs to go from this 3-carbon molecule with a phosphate group to a 3-carbon molecule without a phosphate group. So in summary, we're going from a glucose molecule to 2 3-carbon molecules, which are called pyruvate. And to do that, we use 2 ATP molecules. We get 2 NADH molecules and 4 ATP molecules out of that process. So I was looking at it wrong. We don't use ATP in this process. We're basically making the NADH, and we take that phosphate and make ATP molecules with those. So again, summary, just to make sure I say it correctly, glucose is broken down into 2 pyruvate molecules using 2 ATP molecules, and that in turn generates 2 NADH molecules, 4 ATP molecules. Okay, So the kind of net result of glycolysis is each of these molecules generates 2 ATP and an NADH. Okay, this, let me get my little pointer. This molecule here is referred to as pGal. Okay, and so that's that glyceraldehyde 3-phosphate. Okay, so for every one of these molecules, you end up with this output. Okay, so you have this investment phase where we use 2 ATP molecules, which is this phase, and because we use 2 ATP molecules, we net an NADH, and we net a pyruvate for every pGal molecule. So that ends up being 4 ATP total, 2 NADH total, and 2 total pyruvate molecules. Okay, these NADH molecules will go on to the oxidative phosphorylation step, which is later on. ATP will be stored, so we end up with a net of 2 ATP. Okay, because we had to use 2 to make 4, so we net 2. Okay, so our net cost so far, not cost, but our net gain is 2 ATP total so far. So glycolysis nets us 2 ATP. Okay, all right, so then the pyruvate molecule, so one pyruvate molecule will go on to the next step. And that next step is what's called the prep step. Okay, so with the prep step, the pyruvate molecule is transported to the mitochondria. So remember, glycolysis occurs in the cytoplasm. The prep step takes pyruvate and turns it into acetyl-CoA. Okay, so a coenzyme A comes in, it kicks off a carbon molecule, it makes an NADH molecule, and it ends up being acetyl-CoA. So these 2 carbons with that coenzyme A. Okay, so for the prep step, which is this step here, the pyruvate's converted to acetyl-CoA, produces carbon dioxide and NADH. Okay, so per 1 pyruvate molecule, you get an NADH and a carbon dioxide. Now, that acetyl-CoA will then go on to the citric acid cycle, which is this cycle shown here and also here. So that acetyl-CoA molecule will comes in here, and goes to that citric acid cycle. Now, the citric acid cycle is the 3rd step. So we have glycolysis, we have the prep step, and then we have the citric acid cycle. The citric acid cycle takes that acetyl-CoA and it combines these 2 carbon molecules. The CoA is lost. Then you have these 2 carbon molecules that will attach to this 4 carbon molecule here, the oxaloacetate. Okay, that turns it into citrate, okay, which is a 6 carbon molecule. Then this citrate will undergo a couple steps where it will be converted into a 4 carbon molecule, it will generate some NADH molecules. Okay, this is where your 2 carbons are lost from that 4 carbon. It will make an ATP molecule. It will make an FADH molecule from FAD, and it will make another NADH molecule. Okay, so in total, your acetyl-CoA plus the 4 carbon molecule, that oxaloacetate, so this one plus this one, produces citrate, which is the 6 carbon molecule here. And in total, you generate 2 carbon dioxide, 1, 2. You make 3 NADH, 1, 2, 3, 1 ATP, and 1 FADH. Okay, and so the ATP will be, stored to be used for energy, but then like in glycolysis and the prep step, the NADHs that were made will go on to the last step, the oxidative phosphorylation. So you have NADH, 3 NADHs, and then this FADH2 that will move on. And so remember, this is 1 molecule of pyruvate. Okay, that pyruvate's converted into acetyl-CoA, and then the acetyl-CoA makes these. So this is actually times 2, because you have 2 pyruvate molecules that are made from glycolysis. Okay, so if we look at the next slide, okay, let's clear these drawings. So what that means is we have a net production of 2 ATP and 2 NADH, right? So your NADH here is 1, NADH here is 2. Your net is 2, so let's say these cancel out, so you're left with 2. So that glycolysis gives us this as our net, okay? And then our prep step gives us the NADH per pyruvate molecule. So this is just 1 time, so they're doing this 2 times, so this is times 2 per glucose molecule, right? So we end up with 2 total NADH, and then the citric acid cycle gives us this times 2, again, because we're going to pyruvate molecules, so total of 6 NADH, 2 FADH2, and 2 ATP. So that in turn gives us a total of 4 ATP molecules made so far, 10 NADH molecules, and 2 FADH2 molecules. These will be used for energetic processes, and these will go on to the oxidative phosphorylation step, which is the last step in the cellular respiration process. Okay, and that's this step here. Let me get all these drawings out. All right. So this step generates most of the ATP that we get from respiration. Okay, so remember, we have used 2 ATP molecules here. Okay, and then we'll end up with a total of 38 ATP molecules being made, but some of those ATP molecules were used, so we end up with a net of about 36 to 38 molecules, depending on the efficiency of that of the cellular respiration step. So a real efficient cell can make around 36 ATP molecules, and so most of those ATP molecules are made in this oxidative phosphorylation step. Now, the oxidative phosphorylation happens in the inner membrane of the mitochondria, okay? So let's say that this is the mitochondria. Remember, the mitochondria has an outer membrane and an inner membrane, okay? So what happens here is there's proteins in that inner membrane space that will use the NADH molecules and the FADH2 molecules for energy to move hydrogen protons. So there are hydrogen protons in the inner lumen of the inner membrane of the mitochondria, so this space in here, and it will move them against its concentration gradient. So remember, diffusion, passive, goes from high to low. Active is from low to high. So if there's less hydrogens on the inside than on the outside, needs energy to move those hydrogens. And that's what they're doing. That energy from the NADH and FADH2, the electrons from those molecules are used as energy to move these hydrogens. And so NADH will come in, it will give it an electron. That as that electron passes from this protein to the next protein and the next protein, it will cause protons to move actively out of the interior lumen. Then FADH2 will bind to this protein. The electron will cause a proton to move, and it will go through the transport chain as well. And so because of this kind of chain, and it's transporting electrons, it's referred to as the electron transport chain, also ETC. And this is the only part of cellular respiration that needs oxygen. And you can see it right there, right? Here's our oxygen molecule. And this is actually very important because this oxygen has to be there or it shuts off the electron transport step, and you only can have glycolysis happen when there's no oxygen. And so what happens is this, as the electron gets right here, it will bind with the oxygen and the two protons to make a water molecule. Okay, so this is metabolic water. This is metabolism generating water. Okay, so as those electrons go through, they will, the oxygen will accept that electron, and it combines with the two hydrogens, and it makes that water molecule. If this oxygen is not here, these electrons will build up because they have nowhere to go. If they build up, then the electron transport chain stops. And if that electron transport chain stops, you're not generating those 34 ATP molecules. You actually only glycolysis can happen. So the first part of the cellular respiration process, and we'll talk about that in the last video where we talk about fermentation. And then after these protons have been pushed across into this inner membrane space, now all of a sudden the concentration gradient is set up. So these protons are going to want to move into the interior portion of that inner membrane. And so they'll passively diffuse. And as they do, there's this ATP synthase enzyme here. This enzyme will basically turn like a turbine as these hydrogens move through. You know how like water and wind can move through a machine and cause it to turn and to make energy? Same thing this proton's doing. So as that proton's moving through, it's causing that enzyme machinery to turn. And as it does, an ADP molecule will have a phosphate basically attached to it to make an ATP molecule. And all of the NADH molecules from glycolysis, the prep step, and the citric acid cycle, and then the FADH2 molecule from the citric acid cycle provides the energy in the form of making this proton gradient that will then convert that gradient energy into the ATP energy. And then the cells can use ATP to undergo metabolic processes. Okay, So in summary, the whole citric acid cycle cellular respiration process generates, so after the citric acid cycle, you have 4 ATP, 10 NADH, and 2 FADH. That averages to about 3 ATP per NADH molecule, 2 ATPs per FADH molecule. So that's 30, 34, and then 4 from the point up to the citric acid cycle, so with glycolysis and the citric acid cycle itself. So that means at about total of cellular respiration is about 38 ATP molecules. And then this figure here is just kind of showing that outer membrane, that inner membrane space, the electron transport chain here, so the 4 proteins, setting up that proton gradient out here that then is used to turn the ATP synthase to take that phosphate group, attach it to the ADP to make your ATP. Okay, and so that's, you know, a brief run through of cellular respiration. And then we'll have our final video for this module where we talk about aerobic or actually anaerobic respiration. So this is aerobic respiration because it needs oxygen. We'll talk about anaerobic respiration, primarily the lactic acid fermentation that can happen with glycolysis, and basically how we can generate ATP somewhat in the absence of oxygen, but it's not as efficient as aerobic respiration, where that oxygen molecule is the last acceptor for those electrons as they go through that transport chain like we see there. Okay, so one more video to go, all about fermentation. We shall see you then.